I would like then to start by introducing the Dean of our faculty, Dr. Stan Blade, who will come up and give us a word of welcome. That's very kind.
Thank you, Dr. Grant. It's great to have everyone here for the 17th Bentley Lecture. It's also just so great to see so many people turning out from such a wide array of interests and disciplines. I think I know most of you, and it's really superb to have everybody come to really a celebration. Obviously, that was the intent of the Bentley Lecture, was to bring people together to just poke the curiosity of people and to hear new ideas and new thoughts. I'm really looking forward to Dr. Farenhorst. We had a fantastic lunch with the Bococks, just covering a whole array of territory. As a greeting, I would just say that Faculty of Ales, these are good times for us. Our undergraduate numbers are up. We have well over 500 graduate students doing all kinds of interesting things. If you got here late, please take a look at some of the posters outside. You'll see some of the remarkable research that's going on within our faculty. And just in case you aren't aware, you should also know that our faculty is growing. There are a number of new positions that we've been hiring, but I would just point out 3 or 4. We have just hired an ag entomologist. We're working on a cropping systems position that has been supported by the Western Grains Research Foundation. And we have the 4 crop commodity commissions, barley, wheat, pulse, and canola, that have also supported a position as well. So we'll have 3 around sort of cropping systems 2.0, around a plant microbe, a soils position alongside of ag entomology, and we're also working on really an advanced plant pathology role as well. So it's great to see that plant and crop side of our faculty continue to grow. So it's great to have you here. I certainly wouldn't want to hold up the proceedings from Dr. Farenhorst, but thank you very much for attending. It means a lot to us. Take care.
All right, I would like to take a very brief moment to thank our sponsors, the Alberta Institute of Agrologists, Corteva AgriScience, Alberta Pulse Growers, Bayer Crop Science, Coke Fertilizers, and the Alberta Wheat Commission, who have helped us out financially to make this presentation possible. I would now like to call on the chair of our department, Ellen McDonald, to introduce the speaker.
Thank you, Robert, and it's my pleasure to be here. Welcome, everyone. It's great to see such a good turnout. And it's my pleasure to introduce our guest speaker, Dr. Annemieke Farenhorst. Dr. Farenhorst actually is Dr. Farenhorst. She has a doctorate in physical geography from the University of Amsterdam and a PhD in physical geography from the University of Toronto. She's a professor in the Department of Soil Science at the University of Manitoba. She's also the Prairie Insert Chair for Women in Science and Engineering. She's Associate Dean Research in the Faculty of Agriculture and Food Sciences at the University of Manitoba, and she's the Director of the Insert Create Water Program for First Nations Water and Sanitation Security. So she has a lot of hats that she's wearing. Her research program focuses on pesticides, natural steroid estrogens, and antibiotics in soil and water, and on drinking water quality in First Nations communities. Her projects on pesticide application practices and environmental fate have included projects in Canada, Costa Rica, Nicaragua, and Honduras. She's Canadian representative on the Division 6 Chemistry and Environment Committee of the International Union of Pure and Applied Chemistry, and she's an associate editor for the Journal of Environmental Science and Health, Part B, dealing with pesticides, food contaminants, and agricultural wastes. She's earned a number of teaching, research, and outreach awards, including the Queen Elizabeth II Diamond Jubilee Medal for leadership in pesticide research, and she was named a WXN 2016 Canada's Most Powerful Women Top 100 Award winner. Highly awarded and highly deserving, and we're most pleased to have her with us as our speaker. So please join me in welcoming Dr. Farenhorst.
Yeah. And then it's that one. There. Okay, great. Thank you. Yeah, hello, good afternoon, everyone. I'm delighted to be here. And so when I left Winnipeg, it was minus 53 with the wind chill. And so I arrived in Edmonton, and it felt quite tropical. So for that reason alone, I'm really happy to be here. So I'm originally from the Netherlands, so you may detect an accent. And to be honest with you, I never realized I had an accent until I did hear myself taped. And I thought, oh my goodness, I just sound like all the other Dutch Canadians. But anyways, it is what it is. And this picture, I always like to put a picture on the front cover that represents something. This picture was taken quite some time ago, but I selected it because you actually can see how well I can steer the canoe. So I have well adapted to Canadian conditions. So I'm going to talk to you today about sustainable agriculture, but more so about pesticides. And I'm going to show you some examples of the research we have done and looking at where we detect pesticides, what kind of pesticides we do detect, what are the concentrations we detect, and so forth. This is sustainable agriculture, and when I think about sustainable agriculture, I'm really thinking about economic profitability, I'm thinking about a healthy environment, and also I'm thinking about socio-economic equity. And we not only want that for the short term, but also the long term. And I think sustainability is really a responsibility of all of us, so it's not just the growers, but it's also the food processors, the distributors, the consumers, and so forth. So I think really when we're thinking about sustainability, it's something that we all should own and contribute to. When I think about equity, I have a chair for women in science and engineering. And so one of my roles is to feature women role models. And when I think about farming, what I often see in the farm magazines and the farm newspapers are advertisements that often feature the grandpa, the father, and the son. And then I think, where are the women in these pictures? And so one of the things I think when we're thinking about sustainability and being inclusive in doing so, I think it's also important to realize how many female farmers there are in the world. There are about 30% of the farmers in Canada are actually female. And I've been in countries in the world that this number goes to 40, 50, sometimes 60% in some regions of the world, that the farm activities are actually done by women. So when I think about sustainable agriculture, I think there's a real need to tell the stories of the female farmers and also feature the female farmers in the advertisement that we see in our farm magazines and newspapers. And at the University of Manitoba, I run a mentorship program that is for agricultural students who are female. And they're paired up with mentors. So for one mentee, for one student, there's two mentors. And the mentors are about 50% are from the industry. The other 50% are from academia, not-for-profit, and government. And it's always very nice to run these mentorship programs because you are in a room full of 60 women. And it's amazing how much chatter that can go on if you have 60 women in a room. So I really enjoy running this program, and that's funded by Manitoba Agriculture, so I'm very grateful for their funding. Some of you may want to go to Calgary on March 11th and 12th. That's actually an agriculture conference that is focused on women in agriculture. And it's run by Iris Max, and I've been there several times. It's a very enjoyable conference. It's very inspiring. And you don't have to be female to go there. Any gender can go there. So I would suggest if you have an interest in agriculture, come to the conference, and I can promise you would have a good time there. Lots of inspiring talks that are going on, and usually there are two conferences, one in the East and one in the West. So sustainable agriculture, it's always interesting to be working and presenting on pesticides. So I do also work on First Nations communities. So we're looking at the quality of drinking water in First Nations reserves. And we do find bacteria in the drinking water, which I find very alarming. And I not necessarily always get a reaction from people in the crowd. But whenever I present on pesticides, there always seems to be a reaction, either a reaction that is totally against pesticide use or a reaction that is totally for pesticide use. I'm born in October, and I don't know if you know the signs, but October means that you are most likely a Libra. And so I tend to be in the middle of things, not at one extreme, not at the other extreme, but in the middle. So that's the type of person I am, and that's the type of scientist I am. So, but if you look at sustainable agriculture and definitions thereof, so there might be one from the European Crop Protection Association, that says that the use of crop protection technology, such as pesticides, are important if we actually want to have a sustainable agriculture to feed the world. There are also other opinions, and one that you may have heard of more recently is the regenerative agriculture. Now, that's not a new term, but it's more increasing in the news, and this means that we're looking at rebuilding the soil, which is always good. I'm a soil scientist, so healthy soils, I know the importance thereof. It also means that we may be able to use pesticides in these systems, but there are also philosophies within the regenerative agriculture that we should not be using pesticides. So there are lots of opinions out there, but all of the opinions are actually that this is sustainable agriculture. Now, what I think is, however you look at sustainable agriculture, I think one thing that everyone can agree on, whether you like pesticides or not, is that sustainable agriculture should protect resources, water resources. So whatever angle you look at, that is something that we all agree on. We have to make sure that we protect our water resources. And there is one simple reason, which is we don't only want a healthy environment, but if we pollute our waterways, water treatment may become more difficult and more costly. So it makes sense. Healthy environment means also that we have less cost in terms of providing for clean drinking water. So in terms of pesticide use, this is a picture that looks at annual pesticide use in selected countries around the world. And this is the top 10 countries, and if you look at the very large dot there, that's China. So if we're actually looking at pesticide use, the dominant user, so to speak, is China, but Canada is certainly also in the top 10. And Canada, we have a very large land mass. We have a very large agricultural land base. And so when we actually looking at the application rate per acre or per hectare, it tends to be much less than in some of the other countries. So for example, I worked in Costa Rica, and the applications on an annual basis are much larger than we would have in a country such as Canada, in part because they produce more crops in a given year, but also because they have more insect pressures and so forth. So although we are in the top 10 of pesticide users, because we have such a large land base, the amount of pesticides that are applied per unit area tends to be relatively low compared to many other countries in the world. Now, pesticides are not a new thing. And if you look at the first written record of pesticide use, I don't know if you recognize this person. If you don't, I thought I would give you a hint. And this is actually Homer, and Homer is believed to have written the Odyssey. And if you read the Odyssey, I don't know, anyone has read the Odyssey here in the room? Oh, definitely put it on your to-do list. Read the Odyssey. So if you read the Odyssey, there's a line that talks about the application of sulfur in the hallways. And the suggestion is applying sulfur to deter or decrease the rodents in the hallways. So that was is believed to be the first record of pesticide use. Now, when we're thinking about pesticide use in the history of time, from about, let's say, 1850 to about 1930, 1940, we tend to use inorganic chemicals. And this is a lovely picture from the internet. I'm not sure whether it's actually lovely because it's quite a significant spray there, and you also notice that the person doesn't wear any protection. And so things have changed. Farmers wear protection, in Canada at least. When I was working in Central America, I did see many people applying pesticides without protection. And often when you look at the people's hands, they're full of white residues because people don't wear gloves, etc. In Canada, farmers are well protected. They're very well aware of that. But this is an older picture, and this is an application of lead arsenate. Perhaps you know it's very persistent. These inorganic chemicals are very persistent in the environment. And so what happened at that time, there was a buildup of these inorganic chemicals in the soil to the point that we actually got toxicities in crop production. So farmers started to notice a decrease in the yield. There were incidences about toxicities of people eating grapes that were sprayed or apples that were sprayed, etc. So there was an urgent need to come up with better solutions because, quite frankly, people realized that at that time that the use of inorganic chemicals were not sustainable. So this guy came along, and this is a molecule. This is DDT. And we all know that DDT has been overused to the extent that it did create significant problems in the environment. But at the time, so we're now around the 1935, 1940s, when people were looking for alternatives to inorganic chemicals. This person, Paul Muller, discovered the insecticidal properties of DDT. Now, DDT itself was known for a long time. It was synthesized by a student in the lab in the late 1800s, and it was believed to be useful in making dyes. And so Muller, he was a chemist, and he was screening many, many different compounds, and he was looking whether these compounds were effective in killing insects. And what they were interested in were things like flies and mosquitoes, etc. And he found DDT to be extremely effective. So you can see it kills the fly, it kills the mosquito, kills the louse, kills the flea, etc. And at that time, we had the Second World War, and DDT was actually extensively used by the army, partly in the protection of civilians against certain diseases that were transmitted by vectors such as these particular insects, and it was also used for the soldiers. And it became so popular that Winston Churchill actually mentioned, or he called DDT the miracle white powder. So that's how DDT was celebrated, if you will, at that time. Sometimes when we discover something that we think is useful, we tend to overuse it. So there were advertisements about that women should, at that time, women were typically doing the household chores. And there were photos there of women, and you had to spray the DDT on your clothes and in the couches and so forth. We sprayed it with spray planes, and I have to say there was a very large overuse of DDT, and as a consequence, problems arose. Now, one of the problems why DDT was causing environmental effects and impacted organism is that DDT was very persistent. So we not only applied large amounts of DDT in all sorts of applications, but the DDT stayed around for a long, long time. So DDT, if you look at its half-life, it's 6,200 days, and that's its half-life. And half-lives of pesticides follow first-order kinetics. And what that means is that the graph, if this is day 0, it goes sort of like that. So it's not a linear degradation, it's a first-order kinetic. So if we're saying that the half-life is 6,200 days, it means that it actually stays around for a long, 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 long time. If we compare that with the half-life of the chemicals that we use today, with the pesticides that we use today, this is a graph that we made with over 100 current use pesticides in Canada. And you can see that most of the pesticides that we use have a half-life of about 160 days or less. So anywhere within the 0 to 200 days. So typically, the pesticides we use nowadays are different in the way that they are less persistent than DDT. We still detect DDT in some environmental samples, while we haven't been using it since the 60s. So that's how persistent it is. So when we look at pesticides in the environment, I work quite extensively with Claudia Sheedy, Dr. Claudia Sheedy, who is a research scientist with Agriculture and Agri-Food Canada. And we typically look at about 170 pesticides, sometimes less, sometimes around 170. About 40% of those are current compounds, and about 60% are compounds that used to be registered but now deregistered. So only 40% of what we are looking for are actually that we currently use. Now, why are we interested in looking for pesticides that we no longer use? Well, we're interested if they have stayed around in the environment, such as DDT and metabolites. Most of the pesticides that we look at are either herbicides or insecticides, and that's partly when you look at the method development of looking at pesticides in environmental samples. In the early days, it was heavily focused on insecticides, so methods started to be developed for insecticides. Then we switched more to herbicide use, and the methods were developed for herbicides. So generally, if you're looking at pesticide monitoring, you rely a lot of methods that have been developed over time. So now, if you there is a database which is called the IUPAC Footprint Database, and IUPAC is the International Union of Pure and Applied Chemistry. So they maintain the database, and it's online. If you Google IUPAC Footprint, you can see the database. And if you count the number of pesticides in the database, and I actually did, you will get 1,691. If you don't get that number, let me know, because then we recount. But you may wonder, well, if you only look for 170 or less compounds, are you not looking for a needle in the haystack? Well, the answer to that is that the compounds that we tend to look for, that 40% that is currently used, tend to be the compounds that are used in Western Canada. So we really select these compounds because we know they are typically applied. One of the things I think what is still not forthcoming in science is that we often look for the active ingredient only. And the active ingredient is the ingredient in the pesticide product that kills the pest organisms. So a pesticide molecule, an active ingredient, over time it's broken down, and it's broken down into what we call metabolites or transformation products. Sometimes these transformation products are less toxic, other times these transformation products are more toxic than the original compound. If you look at the scientific literature, most of the information that's published and available really focuses on active ingredients, much less so on metabolites. You may wonder why that is. Well, partly because metabolites are quite complex. So for a given pesticide, there might be 10 different metabolites. Sometimes we're not sure what kind of metabolites might be formed. And when we're thinking about pesticide registrations, we often focus on the primary metabolite only and not maybe some of the others that might be formed. So traditionally, the methods that are available are designed for the active ingredient and not for the metabolites. Ultimately, and I think this is very important to tell, so the inorganic chemicals, what I talked about that were used prior to the 1940s, they are very stable in the environment. What happens with current pesticides, the synthetic organic chemicals, an organic chemical is actually ultimately mineralized, and what that means is that it's transformed into CO2, water, and inorganic chemicals that are relatively benign and are used often as food sources by things such as microbes. So the pesticides that we use, one desire that we would have in developing pesticides would be a pesticide that does do the trick, it kills the pest organism, and then is quickly transformed into and then mineralized. So that's sort of the desire of the chemicals we use. Oops. Now, one thing that I think is really, really important to stress is that the detection limit of pesticides have decreased significantly. So we used to measure at a ppm level, microgram per liter. Then we went to the ppb level, and now we are pretty much in the ppt level, and PPT is nanograms per liter. So we used to look for chemicals, and we could only detect them when they were present at relatively high concentrations in the environment. Nowadays, we're measuring PPT levels, which is a very, very, very small amount. Sometimes people wonder, well, what is a small amount? And I did some calculations, and I came up with this one. I don't know if you know this cave painting, but it is in France. And someone painted this 32,000 years ago. So that's a long, long time ago. Now, if you would say, what is PPT? PPT is actually 1 second in 32,000 years. So let's say we're 32,000 years back. I'm doing the painting. And we go all the way to now. That's how much a PPT is. So it's a very, very, very small amount. So what we are interested in, and when I say we, I typically mean my students, my postdocs, my collaborators. So this is really a team effort. Studies, you know, I mean, you have to do the field work, collect the samples, you have to process the samples in the laboratory. So there's quite a bit of work involved in pesticide analysis. So one of the things we are interested in is if we sample different rivers that are in locations where we have agricultural use, where we have perhaps urban use of pesticides, what type of pesticides and what kind of concentrations do we detect, versus if we go to rivers that are perhaps more in forest areas, wetland areas, etc. So this is a study we did in 2016 and 17, where we went to the different rivers and sampled along those rivers and looked for the pesticides that were potentially present. So this is a top 3 pesticide present in the Manicouagan. I know I always get my tongue twisted in that pronunciation. My students are very good in pronouncing this because they're presenting on it all the time. But this is the river that you saw more in the northern location, so it's flowing through a forest area. We do detect pesticides at these locations, 2,4-D, MCPA, and bentazone. And these three chemicals have something in common in that they are all herbicides and they have a high water solubility. So these chemicals we detect, the frequency is around 25% to 37.5%, depending on the chemical we looked at. And the maximum limits are indicated in PPT. So we have a maximum concentration that we detected across those 2 years of 591 nanogram per liter for 2,4-D, 61 for MCPA, and 78 for bentazone. Now, I put on a reference, and I could have looked at other organisms. I got this number from that IUPAC database, which is really seen as the the best database out there when it comes to pesticide, environmental parameters, or toxicology parameters associated with ecotoxicology. So I picked a fat minnow, and a fat minnow is typically used in screening of toxicity of chemicals. And just to give you a number, so from the database, the acute 96 hours LC50, so this is a lethal concentration that would kill 50% of the organisms that are exposed to that particular level. The LC50 is 100 million nanograms per liter. So what we find in the environment tends to be much lower than this particular parameter of toxicity, which is in this case the fat minnow. Now, we do, however, find that when we're looking at areas that are having agricultural land surrounding it or urban area, we do find that typically the frequency of the detection of compounds increases, as well as the concentration of the compounds that we detect. So MCPA and 2,4-D, and I highlight these chemicals as examples because these chemicals tend to be the top chemicals that we detect regardless of the river where we are sampling. MCPA and 2,4-D are phenoxy acids, so these are chemicals that are widely used, particularly in Western Canadian agriculture, the prairies. So these are chemicals that are quite widely applied, so it's perhaps not a surprise that we do find them. They're used in agriculture, in urban type of settings, as well as in the forest industry. So we find different concentrations, the Red River, and then we also sampled the Fisher River, and we got a high spike of MCPA, and that was at location R2 that is indicated there on the slide. The high spike, sometimes you get high spikes and you want to look at the concentrations of all of the other pesticides in the sample to make sure that it wasn't a faulty analyte in the laboratory. In this case it was not, so we clearly saw a peak of the MCPA, but not of some of the other pesticides that we found in the sample. So this could have been perhaps a spill of some sort. It's always hard to say where the chemicals come from directly. And most of the pesticides that we find in the environment, we do not necessarily know where they're coming from, unlike some of the other things that we have experienced, such as when there is a massive spill and everyone knows that spill has occurred. So we're not sure, but we suspect there was some kind of perhaps a spill of the MCPA. And just to let you know, so we sampled about every 2 weeks, and so 2 weeks later, this concentration was much, much lower. Now, the interesting thing about MCPA and 2,4-D is that they also travel in the environment, so they can volatilize. into the atmosphere, and then they are transported. And I think this is probably one of the best examples that demonstrate that. So this is a paper I contributed to, so the data that we looked at was concentrations of MCPA and 2,4-D and a host of other chemicals that are described in the paper. But we looked at the mean concentration, the maximum concentration, the detection frequency, and so forth. And so these are in wetlands, prairie pothole region, where a lot of wetlands are integrated within our agricultural land. And if you're looking at the minimum till farms, MCPA detected 100% of the time, 2,4-D detected 97% of the time. So that's in minimum tillage farms. If we're looking at organic farms, so this is sampling wetlands that are situated on organic farms, no pesticide, no synthetic pesticide use. The MCPA, 100% detection, and 2,4-D also 100% detection. So there is really no other explanation for that unless the organic farmers all use pesticides in a secret, but I don't think so. There's no other explanation than that these things are traveling in the atmosphere, and then we have settling dusts that are settling in the wetland, or we have a rainfall that carries some of that dust and gaseous pesticides to the wetland. So we definitely know that even if we are in pristine environments, sometimes we find pesticides because they do travel. This is an example of how we can measure atmospheric deposition. And if you look at that picture there, it's a 1x1 square pan, and it's slightly sloping, and there has a little hole, and so the water drains into a very, very large flask. It's the same flask that you can use to make wine. We literally use those, but we don't make wine. We just collect the rainfall. And the dust also settles on this pan. And then when we collect the sample, we always rinse the pan so we're sure everything is in there. And we then collect the sample on about weekly basis. Now, this picture is not from an agricultural field, or the graph is not from an agricultural field. This graph is actually from a study that we did about 5 years ago in the city of Winnipeg. So we had these systems set up in people's gardens because we were interested in what kind of pesticides can we find in the atmosphere in an urban environment. And so we collected 36 samples in total. Every sample we collected, we found at least one pesticide. And I put there the red line just to as a reference. So if you look in 20% of the samples, we detected 10 different pesticides. So when we're thinking about the atmosphere, there's a lot going on, and there's certainly a number of pesticides present. Now, going back to the Red River, Fisher River, Winnipeg River, and the difficult to pronounce river, the number of compounds usually are highest when we have areas of agricultural land and urban environments. Detection frequency increases, as well as the cumulative concentrations of the pesticides that we find. Now, this is a picture of the Red River because, sometimes we take samples upstream and then downstream in the middle type of thing of rivers. But in this case, we were interested in looking at more detail at the Red River. So what we did is we sampled at many locations around the Red River, all the way from the beginning where it enters from the United States into Canada, all the way to Lake Winnipeg, because the Red River and all of the other rivers I've discussed so far, they're all entering Lake Winnipeg. And so we also, in this case, sampled some of the tributaries of the Red River. And what you see here, all those sort of grayish bars, those are different pesticides. I just added them all up. And if you can see, so typically the tributaries to the Red River contribute not as much pesticides as what is coming from our friendly neighbors to the south. So if you're looking at going from this way to that way, all the way to Lake Winnipeg, you see that there's some variation in the concentrations of pesticides that we detect along the way. But the key point is, if you're looking at this, really by the time the Red River enters Canada, it already contains a significant amount of pesticides that comes from the States, and it continues to contain the pesticides. So one of the issues when we're thinking about the detection of pesticides in the environment, it doesn't know boundaries, so it goes from one country to the other, or from one province to the other province. And I think we have to keep that in mind. So I've been talking to you about samples that were collected from the water column, but we also collect bottom sediments. And I guess, again, you know I'm using the word we here, but most of this work is actually done by my students, as you can see. So this is Mauli and Marufa, and they are working very hard. And Marufa, here on your right-hand side, she's collecting bottom sediments. Now, sediments are much more difficult to analyze than water samples. So this is an example of the process that you have to take to actually look at pesticides that are present in sediments. And it's quite an elaborate process. So typically what we find then, so this is from 2 different years. We looked at slightly different numbers of compounds, depending on the year and the environmental compartment. Typically, we find 24 to 34 different compounds across those 2 years. And so meaning that a lot of the compounds we actually analyze for, we never detect. So about 85% or so of the compounds we look for never get any detection in 2 years. So compounds that we do detect, and I put in this slide looking at glyphosate because there was a special request to learn something more about glyphosate. And so glyphosate is an interesting chemical in that it is very strongly sorbed onto soil particles. And as a consequence, because it's so strongly sorbed, it's relatively immobile, so it would be very rare to detect glyphosate in groundwater. And so when we're looking at where glyphosate exists in a river system, it really exists in the bottom sediments. We never found any glyphosate in the water across those 2 years. So glyphosate may enter, it gets very strongly held onto the bottom sediments. The concentrations that we do detect of glyphosate in the sediment tends to be much higher than other chemicals. AMPA, or AMPA, is a metabolite of glyphosate. And so glyphosate tends to be present at higher concentrations than other chemicals. You also see DDE and DDD, so we still detect some transformation product of DDT in our samples. Water, if you're looking at what we do detect in the water, it's only of this list of chemicals that we see in the sediment, only 2,4-D and MCPA are both present in the water and the sediment. So glyphosate, we also included glyphosate in our bulk deposition sampling study. So this is back to the study where we looked at what's present in the atmosphere in urban environments, such as the city of Winnipeg. The number one that we did detect in the bulk deposition samplers was the pesticide glyphosate, followed by 2,4-D mecoprop, which is something that is mainly used in urban and not agricultural environment, MCPA, and so forth. So what that means is that, as I mentioned, glyphosate is very strongly sorbed by soil particles or sediment particles. And so glyphosate, when we think about glyphosate in the atmosphere, it's mainly present in dust particles. So this is an example of that same study, the bulk deposition study in the city of Winnipeg. 2010 was a relatively wet year, more rainfall. 2011 was a relatively dry year, less rainfall. The darker bars are the weekly deposition of glyphosate. The lighter bars are the weekly deposition of 22 other pesticides combined. You can clearly see that in a dry year, you get a greater amount of glyphosate in your bulk deposition. And the reason for that is that glyphosate is present in dust particle, atmospheric dust, and hence it can move further distances in a dry year before it's being deposited. So it's very clearly that glyphosate, the way it's present in the environment, is in association with dust, sediment, or soil particles. Now, neonics are always interesting too. People always seem to be interested in knowing more about neonics. I find neonics somewhat problematic because it's so difficult to pronounce all the different neonics, so I'm not even going to try. But this is a picture that shows the neonics that we detected in the river systems. So these ones with a star, these are actually, they might be phased out by Health Canada within the next 3 to 5 years. We did not detect this one at all, and the reason for that is that it has a lesser use in the agricultural industry, but also if you look at the half-life in soil and half-life in water, it's much, much, much lower than the other chemicals. So when we're thinking about pesticides then, if we find them in the environment, it's a function of how much do we use these chemicals and how long do they persist based on their chemical properties. Now, I should point out that we have only focused on neonics in river systems. Other studies at the University of Saskatchewan, they have looked at neonics in wetlands, and we get very different results in our river systems from those that have been found in the wetlands. So when we looked at the neonics, those 3 we detected, we never detected them above 50 nanograms per liter, and we only detected them in water. This somewhat agrees with the studies of the University of Saskatchewan that have been done for wetlands, in that they also only detected neonics in only 6% of the sediment. So it means that neonics in the environment do not necessarily partition to sediments quite readily. It stays in the water phase. But our concentrations are very different. So as it's 50 nanograms per liter, less than, and here we're looking over 1,000 nanograms. So each water system is somewhat different, and I think we have to keep that in mind. So in the last section of the talk, I would like to just make a few comments on mixtures. So I've been presenting things on single compounds, but of course there's also an interest in looking at mixtures. And sometimes if 2 or more chemicals are present, they may become overall more toxic together than if they would be present alone. And sometimes we actually see that if you have a combination of chemicals together, they may be less toxic than they would be alone. So it could be synergistic or antagonistic effects. And so when we're looking at mixtures, we often find mixtures in our river system. So 72% of the water samples that we took over 2 years contained more than one chemical, more than one pesticide. 51% of the bottom samples contained more than one pesticide. If we're looking at groundwater, and this study was actually done in Alberta by Camilla, was here featured here in the photo. If we're looking at groundwater samples, only 3% of the groundwater samples contain mixtures. So overall, when we sample surface water, we're more likely to find pesticides than we sample groundwater. And that's why I love soils so much. Soils are a great buffer. It really retains the chemicals rather than moving them readily to groundwater. So in terms of the mixtures then that we do find in groundwater, and here we go, MCPA and 2,4-D were present in these mixtures in addition to some other pesticides. And what I find fascinating is that so MCPA and 2,4-D are very, very similar structures. So MCPA has here a methyl group on the position 2 of the ring, while 2,4-D has a chlorine. So otherwise identical structures. Now, what we actually found that the MCPA mixtures, all the MCPA mixtures, were always detected in the spring or the summer in the groundwater. However, the 2,4-D mixtures were mainly detected in the fall samples. So what that means is that a very tiny slight difference in the molecule can actually have an impact in the way that the pesticide behaves in the environment. So 2,4-D and MCPA are likely to be applied at the same time. They're sort of interchangeable in the cropping systems. And so what we do see then that 2,4-D is most likely to be a little bit more strongly retained than the MCPA and perhaps a little bit more persistent than the MCPA. And as a consequence, we detect them at different times in the groundwater samples. So how do we know drinking water is safe? As soon as we start talking about groundwater, we often wonder about, is it safe for us? A lot of us rely on well water. And so actually, well, how do we know it's safe? Well, actually we measure when it's not safe. And so these are some examples. If we would look at maximum acceptable concentrations, glyphosate, 280,000 nanograms per liter, 2,4-D, 100,000 nanograms per liter, and bromoxynil, 5,000 nanograms per liter. So I'm just providing a range of examples to you. Now, if we're looking at, so these are under the regulations established by Health Canada, the Pest Management Regulatory Agency, and these numbers of the maximum acceptable levels are very different if we, for example, look at Europe. Europe has a very different system. This is to wake you up, by the way, this bright light of blue. If we're looking at the European Union, their regulation for any pesticide, it's 100 nanograms per liter. If we're looking at a mixture of pesticides, according to the European Union, the maximum guidelines are 500 nanogram per liter of all pesticides combined. So I'm flicking back to what we have in Canada. These numbers are drastically different. Now you may wonder why that is. We are stronger as Canadians and we can withstand more than the Europeans. But no, that's not the case. The reason why this is that we have very different regulatory systems. So in Canada, we base everything on toxicological data. So we look at data that has been derived from animal studies, so rats, mice, rabbits, etc. And then we do a whole bunch of calculations. We put in some extra safety factors, and then we have a particular concentration that we define as the maximum acceptable concentration for humans to consume. Europeans have taken a different approach. They have said, if we can detect a pesticide, it's not good. And if you're looking at sort of the traditional technologies, the 100 nanogram per liter is about the concentration of any pesticide that you can detect using the technologies at the time that the regulatory numbers were developed. So it's a very different system. I don't think one system is better than the other. I think Europe is maybe a little bit too conservative, but there are differences. And so if we look at some of the concentrations we find in the groundwater, according to the Canadian guidelines, you can readily drink the water. If I would comply with the European guidelines, I would not be able to drink that water. So lastly, I always think it's important for us to think about what can we do to minimize pesticides moving to water. And so this is an application I think is something that we really should explore. This is called biobeds. And so what that is that when farmers have applied the chemical, when they rinse out their tanks, you can actually put that rinsate there on that grassy area. And under the grassy area, there's a big pit that contains what you call a biomixture. And the biomixture may contain soil, peat, straw, etc. And it's designed then to capture the pesticides that are in that rinsate. And because we have a lot of organic matter in that biomixture, it's good for microbes. They eat and they grow and they degrade those pesticides, because most of the pesticides are actually degraded by microorganisms in the environment. This is something that was invented by a Swedish farmer decades ago. I think anything that's invented by farmers is often a good thing, because, you know, the farmers saw a need for this and developed that. It's when I worked in Central America, I saw this at a wide range of farms. It's quite popular there. It's used in Europe, in many different countries in Europe. Just recently then in Canada, particularly Dr. Claudia Sheedy, who I talked about before, was at Lethbridge with Agriculture and Agri-Food Canada. They just produced a whole guide on how to establish these biobeds. And I think that we really should explore this, and because I think it would be extremely helpful in protecting things such as groundwater. So with that, speaking of straw, I hope you enjoyed this presentation. These are some of my lambs that were born this year. And I also want to thank the sponsors that sponsor all sorts of programs that we run. I run a lot of outreach programs and workshops, and so we have a variety of sponsors, and we are very grateful for that. And lastly, these are some of my students, and I really am extremely grateful. Students are wonderful to work with. They keep you young. And they're always working hard, so I'm very grateful for them. So thank you.
Okay, thank you, Annemieke. I'm sure there'll be some questions. Yes, go ahead.
Alberta, Wheaton Barley. Thank you, that was awesome. I have two questions.
You're limited to one, right?
Would you be able to go into detail about how you determine where you're sampling from and what characteristics you look for in sampling sites?
Yeah. Yeah, that's a good question. So it might be a variety of reasons. So the Winnipeg River, for example, we wanted to sample before and after some hydro dams. So we were interested in looking at the pesticide concentrations before the hydro dams and after the hydro dams. So that could be one example. For the Red River, what we did if you remember that intensely sampling points along the Red River, we set that up because initially we wanted to look whether there's a potential contribution of golf courses to adding concentrations into the river. So typically we design a study because we have certain questions in mind, and that's how we select our sampling sites. Sometimes we put a sampling site at stations that are monitored for water depth and velocity. So we may choose to have a sampling location near that site, so we actually have some further information about the water flow, etc. So there are really a variety of reasons. To give you a very silly reason, in Winnipeg, when we installed the bulk deposition samplers in Winnipeg, we installed them at the homes of the graduate students or the secretary of the Department of Soil Science. So give me a whole host of reasons. Yeah. You want to ask your other question?
I think I saw it.
Oh, sorry.
Are there more questions? Anyone? Oh, no. Oh, sorry, yeah, I didn't quite see. Yeah, thank you, go ahead.
I've read research that connects glyphosate with celiac disease, and there's a veterinarian in Saskatchewan, I won't give his name, but he's been courage enough to suggest that glyphosate is affecting the digestion in cattle and making them sick. What is your opinion?
My opinion? Well, yeah.
Sorry, yeah. Sorry, I'm a little distracted by some noise. Okay, all right. Sorry, it was a question about the association between glyphosate and celiac disease. Is that?
Yes, and also sickness in cattle.
Sickness in cattle.
Problems with the gut.
Okay, great.
Yeah, so my opinion, my first opinion is that with any pesticide, there's a wealth of opinions and information out there and claims out there. So this specific one, I to be honest with you, I wasn't aware of. What I know of glyphosate, if we're thinking about its retention onto organic matter, it's not very strong. Glyphosate, it has a phosphate molecule in its own molecule, phosphate group. And so it's very strongly sorbed to things such as iron oxide, soil particles, rather than crop residues. You know, if a cattle gets sick, one of the questions is how did the cattle ingest the glyphosate? So that would be, you know, one thing I would be questioning, or not questioning, but, you know, would like to discuss in. I think there are a lot of claims out there. I think, you know, sometimes we hear about things. I think if someone claims that, we really have to look at the overall literature that's available. If it's a claim that is found by a number of people and scientists, you know, I think we have to focus on that. I not necessarily think if it's a claim by someone, because I've heard a lot of discussions about any chemical. Is this related to that? Is that related to that? Is that related to that? I think we really have to, if someone thinks that or believes that, I really think we have to either do a sophisticated study if there's no information available, and I mean a scientifically sound study, before we actually can say, yeah, that's true or not true. And I think that's often what's missing in a lot of claims. I think often what's missing is a scientifically sound study, that everyone agrees this is a good design, the answer that you get is based on scientific evidence.
Oh, here are more questions. All right. I saw, is that Bill in the back?
Yes.
Yes.
So
Go ahead.
thank you very much. I enjoyed the presentation very much. A couple of things caught my eye. I will have many questions for you later. But the first one, I really, I don't want to say shocked, startled. The glyphosate in the air in cities. The concentration to me looked high. So 100, 200 micrograms per square meter. I live in a city, so I think about this a little bit. So the question is, did you look at the particle size distribution of the aerosols? In which size fraction was that glyphosate?
That's an interesting question. So the way these samples are collected is in that 1x1 stainless steel bed that has a little bit of an angle. And we leave it out there for a week. So the numbers that you see are a cumulative amount in that week. So we would collect rainfall during that week, settling dust during that week, and then we rinse it before we take that big 20-liter bottle and then take subsamples for further analysis. So we didn't really separate the particle or the dust from the water. The graph that you saw, that in 1 year there was a drier year and there's a higher amount of glyphosate in the bulk deposition. We don't really separate the water phase from the particles. We only know the bulk, the total amount. And so when we look at one dry year, that is a higher amount of glyphosate than a wet year. We imply that that is because of glyphosate is moving more in the atmosphere on dust particles. Now, in terms of the numbers, sometimes the numbers sound high, but again, we're looking at nanograms per liter, so we're looking at that 1 second in the 32,000 years. If I would have put the numbers in ppm, the numbers would have looked much smaller. So I don't necessarily, I think it's much higher than any other chemical, and I do think that's a reflection of the frequency and or the extent of use of glyphosate. Typically, when you're using it, you're likely to find it more readily. So it's a reflection of that. Whether it's a high concentration in terms of, impact, I don't think so, but I do know that sometimes people were concerned about that and asked me, should I be concerned? And I always say, if you are concerned, make sure that the water bowl you might have for your rabbits or your cats or your dog in your garden, you may want to just, freshen it every day. And if you are concerned about what may be entering on the toys that your kids play with, well, maybe you rinse it out if you're really concerned. I'm not awfully concerned as a scientist, but I do understand that some people are concerned, particularly if you're more sensitive to it. I also recognize that on average, some people are not as sensitive as some other people. So if you are concerned, there are things you can do, such as rinsing.
All right. I saw another hand. Oh. All right. Elizabeth, is it? Yes. But please call out loudly.
Yes, sorry. So I got to spend the morning with the city committee looking at pesticide policy update. So you have 10 precious days to implement city policy if that interests you. So you would just need to go to Edmonton.ca pests for a survey, or I'll write it on the board if I can find a pen where you can send bigger opinions in. But they have not changed the policy for 14 years, and I'm sure you've heard about chlorpyrifos and other use of chemicals. So if you would like to influence that, now is the time.
Thank you. Other questions?
Over here.
Sorry? Oh, yes. Okay, go ahead. Yes.
Thank you very much. It was an interesting presentation. I'm an organic farmer from central Alberta, and I noticed your slides were of permanent wetlands. So in my area, there are ephemeral wetlands. And zero till now, of course, is 4 herbicide applications a year. And with 150-foot spray booms going over, I see a lot of the wetlands or ephemeral wetlands are sprayed now. So my question is on the pond critters and the trophic food web.
Yep.
And if you've done any work there.
Yeah, so quite some time ago in 2010, and this picture that I show, yeah, it was one of the wetlands, but we also have looked at ephemeral wetlands versus permanent, and so one study we did, and again this is 8 years or so ago, we chose a permanent wetland and an ephemeral wetland, and what did is we mixed a whole bunch of chemicals together, mainly focusing on MCPA, 2,4-D, etc., and then we oversprayed it. So we did exactly what you're asking. We pretended that there was an overspray, and I still feel a bit bad about it because I'm actually contaminating a wetland. But what we were interested in is following the fate of the pesticides, and we also looked at organisms, mainly invertebrates. What we found was that we actually saw an increase in the invertebrates after the overspray. And the reason for that is that it probably killed off some of the organisms that the invertebrates are feeding on. So it was kind of surprising for us and we never actually published that study because the PhD student was working on that, had so much other data that he didn't need it to finish his thesis. And then he promised to write up that paper, but he got a job and then we never got around to it. Happens a lot. But it actually showed that there was no negative effect, and I don't think there was a significant beneficial effect, but we certainly saw a little increase in the invertebrates. So that's how far we have studied that. I do recognize that there's a difference between permanent and ephemeral. One of the things I would be interested in is because a lot of the chemicals do partition to sediment. I've not done those studies, but I would be interested in doing that, is actually looking at ephemeral wetlands, sample the bottom sediments over time as they dry up, and then looking what's the fate of those pesticides that actually are in the sediment layer. I think it would be kind of interesting, and the same scenario, maybe we have an ephemeral wetland which is dry in a given year. The farmer uses that for the crop production because you want to maximize your land area. If you do that, and then with the following year is a wet year, and the ephemeral wetland fills up with water, are these chemicals that you sprayed in the previous year released to the water column? I think that's fascinating studies. If you have funding, let me know. I will be there.
Yeah, if you're hiring, maybe.
All right. Yes, go ahead.
I had a question about the mixtures, and you're mentioning that the chemicals and the pesticides can have synergistic or antagonistic interactions, and then thinking about your graph how with aerial deposition there was, you see more of it in the air and land. Have you ever looked, or has anybody looked at, say, interactions between chemicals from wildfires and then pesticides in the air? That you imagine sort of in a dry climate, you have all these pesticides in the air or whatever, and interactions that might be happening with things that are coming off the fires.
Yeah. Interesting question. I know of studies that have looked at the environmental concentrations of a host of chemicals, PAHs, etc. I'm not aware of any ecotoxicology studies in sort of mixing it in that way. I think in terms of pesticides, there are not enough studies on looking at mixtures. One of the things we have done in terms of looking at mixtures, we have looked at phytotoxicity. So this was group 2 herbicides. So we looked at single compounds and then mixing compounds and see how toxic it is to plants. Both from the point of view of weed control, but also from the point of view of carryover from one year to the next, and they may affect sensitive crops. In that particular study, we actually found antagonistic effects. So when you combine them, the phytotoxicity was less than you would expect based on if you looked at them individually and then do the calculations of how toxic should they be if you combine them. So our experimental toxicity was less than you would expect if you do the calculated combined toxicity. I'm not aware about any elaborate study that would look at different types of chemicals. And partly I think the challenge is what mixtures are you actually looking at? Because, I mean, we're using so many chemicals. We could look at household products, natural chemicals, so to speak, synthetic. There's so many combinations, and I think one of the limitations of studies is really funding. You know, I mean, scientists, you know, get scientists together and they would dream up any study and they would be saying, yes, if you say, do you want to study? Yes, we go, yes, let's do it. But really the limitation is funding. So you know, if there's funding, I can find a scientist to do that work.
All right, I think have we decided who's going to ask the question?
I'm better looking. Do you track, or do you have any future plans to track the chemicals and pesticides used in organic farming that are ending up in the rivers as well?
I haven't made plans, partly because, you know, I mean, you establish collaborations and laboratory equipment. Again, you have to apply for funding. You know, if you're lucky, you get the funding. You get the laboratory equipment in place. So often scientists tend to be specialized in things that they have done for quite some time, and they tend to stay in that direction. But I mean, I'm someone that's interested in a lot of things. I think it would be something that we could look at for sure. Yeah, but I haven't thought about that in particular. Yeah.
All right, we'll take one more question because I see there's one thing still key to ask. Thanks. Are there any more questions in the room? No. Okay. Oh, jeez. All right. We'll take Molly's and then yours and then that. All right. Okay.
Like in some countries, they use lots of insecticide, pesticide, and herbicides because of intensive agriculture crop production system. But then at the same time, I'm talking about India, China, and some other countries. They also burn straw, especially rice straw. So, what is your opinion on the pesticides? They were sprayed onto the crop, and then it's burnt. And what's the fate of those pesticides?
Good question. Two things. In terms of pesticide use, from the work in Central America and intense pesticide use, one thing we clearly saw that the people who are affected by the pesticide use, as in becoming dizzy and residues on their hands, were typically the farmers that receive very little to no outreach from government or the industry. They're often farmers on very small plots, and when I say small plots, it may be a quarter of the size of that site. Very small plots. People don't really know how to apply. They don't have funding to buy protective gear. When I went to sort of more large-scale commercial farms, it was completely different. So it was people being protected, there were safety courses, etc. So I think a lot when you're thinking about high pesticide applications, it tend to be the farmers that we still need to reach in terms of informing them that it's not a good idea to spray chemicals and have things on your hand. Now, in terms of the straw, one thing what happens with the straw, if you get residues, I would think that if you burn the straw, you probably get a greater retention of the pesticides in your soil. If you would have pesticides on the straw, maybe a portion of that volatilizes into the atmosphere. Not sure.
Is there anybody has any information how much goes into the air? How much goes into the soil?
I'm not aware of specific studies that have measured that. And it also depends on when that is happening, because when you apply a chemical, it's being degraded by microorganisms or by abiotic chemical reactions. So if you would apply the pesticide, burn the straw, you probably get a little bit more going up than if you would apply the chemical, you know, you wait for some time and at the end of the season you burn the straw because there may not be anything left on the straw. So I think it depends a bit on the timing, the amount, the type of chemicals that you apply, and whether they actually are being retained on the straw. To be honest with you, we have done studies whereby we looked at the retention of trifluralin in that case, something we also use in Western Canada on different types of straws. We used wheat straw, all the type of straw, and then we compared that to soil. And what we found was that the straw absorbed much, much lesser pesticides than the soil itself. So it can also be that you apply pesticides, and if there's straw, that the pesticides just move to the soil.
Okay, our last question? Go ahead.
Finally.
This is it. So my question is around, like, the biggest theme that I see out of this is the chemistry of that actual chemical and the amount and whether it links with soil or water.
Mm-hmm. Yeah.
So your research on the neonics, or what you presented on the neonics, was that, sorry, Manitoba or?
Manitoba.
Okay.
Yeah.
So given that how much of the relation between neonics and the environment, and the amount of moisture in the environment, and the variance of how much moisture we see in Western Canada, from Manitoba to Alberta, and then from Edmonton to Alberta, and then looking at the PMRA, or sorry, to Lethbridge, and then looking at the PMRA, who's actually trying to, it was looking at whether they want to completely eliminate imidacloprid and clethianidin, which is huge for wireworm in southern Alberta. So do you think there could be some type of middle ground that can be found with this variance of moisture and how much it plays a role in neonics being an issue?
So first of all, I'm a Libra, so I'm always for the middle ground. But what I do, you know, I'm part of the subcommittee of the IUPAC, and one thing I like about that subcommittee is that it has a diversity of people on it. So we have people from governments, industry, and a wide range of universities. And these are people from all over the world. And so we have dialogues and often intense discussions because there's sometimes disagreement. And so when I think about, you know, the way you're looking at it and that it's actually really complex because it depends on the weather from the year to year, overall from region to region. You know, I wish that, you know, we had a model, and maybe there is a model like that that I'm not aware of, but I wish we had a model like that in Canada, like the IUPAC, whereby we have people from the industry, people from a wide range of universities, people from government, people from NGOs, and that we actually discuss these things, and that we are coming, everyone can bring their papers that they think is important, we all read it, we have a discussion, and then help in terms of making recommendations. I think that's a good way to start, whether it's, you know, I mean, not only in the neonics, but I think with many other pesticides that people may be worried about. So that would be my preference, but I definitely think that there might be variations in the concentration depending on what water system you look at, what year you look at, where you sample, etc. And unless we have the full picture, and you usually get a better, fuller picture if you have more people in the room that may have a diverse variety of backgrounds and ideas than if you make decisions with a smaller group that comes from the same standpoint.
All right, I'd like to thank Dr. Farenhorst for an excellent talk and her patient response to your questions.